Good morning, friends. Today I came up with one more small topic about pharmacology of GABA, and this is GABA is a neurotransmitter which is being synthesized in brain, and that means the spinal cord and brain, which are both of them is called as central nervous system. And this central nervous system mainly consists of two parts, as I said before. It consists of brain and the spinal cord. So by this we can know that the GABA is being synthesized both in brain and the spinal cord. So what is actually the main function of a neurotransmitter? The neurotransmitter is something which has been synthesized by the neurons so that the messages has been sent from one place to the other place by the help of axons, and it also may be sending by the action potential, that means nothing but the firing of the neurons. We know that the peripheral nervous system consists of different neurotransmitters like noradrenaline, adrenaline, acetylcholine and natropin. But in the central nervous system we come across the different types of neurotransmitters like GABA, glutamate, 5-HT3, DOPA, acetylcholine and serotonin receptors which are being synthesized by 5-HT3, and some neuromodulators which are called as prostaglandins and endomorphins. And so we have been seeing about different types of neurotransmitters which have been synthesized in the brain, and out of which only two can be taken into consideration, so that one is excitatory, the other one is inhibitory. And according to this, the GABA is always called as an inhibitory neurotransmitter. That means it is inhibiting the excitation which is created by neurotransmitter called as glutamate. And here we can see that whatever the excitation as the glutamate has been synthesized for the firing of the neurons, whenever there is more stress, whenever there is more emotional feelings, whenever there is disturbances of behavior which is related to the limbic system and also the temporal lobe and also amygdala, where the emotions has been not be controlled and the glutamate is more and more excited and the firing of neurons have been happened, at that place the GABA comes into the picture. And what is the function? As I said before, GABA is always an inhibitory neurotransmitter, and today we will see what is GABA. GABA is called as gamma aminobutyric acid, which is being synthesized in different parts of the brain, and one is called as limbic system, which is controlling your emotions and feelings, and also it is being synthesized in the areas of eyes, the temporal lobe and amygdala, which is all the three are being related to the behavior. So what we can see that the GABA is one of the neurotransmitter which is doing two main functions. What it is controlling your emotions and feelings, and it is controlling your behavior. We can know here that it is also synthesized in the eyes. Why? Because whenever there is some stress, whenever there is some emotional imbalance, we try to tears ourselves. So in order to relieve some stress, the tears may be helpful so that the stress of the body will be controlled. And so we can see that the GABA comes to that place so that it has been inhibited at that area, and the emotional balance between the eyes and the other body systems are being controlled. So we can say that the GABA is a neurotransmitter which is maintaining two themes. One is called emotions and feelings and behavior. So we can see that it is emotions and feelings versus behavior. So whenever there is a imbalance in the behavior, the emotions and the feelings try to upgrade itself, upgrade outside. So in order to control your behavior and in order to control your emotions and feelings, the GABA system released at the place of limbic system comes into picture. And as I said before, GABA is one of the neurotransmitter which has been released in the brain areas, particularly what is shown in the behavior. And now we'll see what is the synthesis and how this GABA has been synthesized. And main we have to know that it is being called as amino. Nothing but it is being synthesized from the amino acids, and one of the amino acids is called as glutamate. Here one of the main cycle which is being involved is called as Krebs cycle. Have you indicated over here? The Krebs cycle is one of the cycle which has been having some compounds called as alpha ketoglutarate, succinic acid, and different types of compounds over there. But the GABA is being synthesized from different types of amino acids. Particularly, it is collecting alpha ketoglutarate acid from the Krebs cycle, and it is being converted into one of the amino acids called as glutamic acid. And by it is done is by an enzyme called GABA T. What is GABA T? GABA T is being indicated as GABA alpha oxyglutamate transaminase. So we can call simply it is as GABA T. Now, with the help of this GABA T enzyme, the alpha ketoglutarate acid collected from the Krebs cycle is being converted into glutamic acid. And once this glutamic acid is being synthesized in your presynaptic cleft and the postsynaptic clefts, we can see that it is being converted itself into GABA. What is GABA? I said before, it is called as gamma aminobutyric acid. And this glutamic acid is converted into GABA by an enzyme called GAD. What is GAD? GAD is called as glutamic acid decarboxylase. We can see that it is doing carboxylation actions. So once we can see before itself, the alpha ketoglutarate acid taken from the Krebs cycle is being converting itself into glutamic acid, and this glutamic acid is converting itself into GABA, and this GABA is done both of them by GABA T transaminase and also decarboxylase of glutamic acid. So we can see that the GABA has been synthesized in the presynaptic clefts, and you can see one more thing, two semicircle circles like this, circle-like structures. So what are these? These are called as synaptic vesicles. These synaptic vesicles which have been present in the presynaptic area are these being collection of your different types, many of your semisynaptic circles, not actually semisynaptic, it is synaptic vesicles, and these synaptic vesicles are being taken and become the storage place of your GABA. The synthesized GABA from your glutamic acid is being stored into the synaptic vesicles, and it always stays in the presynapse until the firing of all the emotional balances and the emotions and feelings, and also the behavior has been changed. It stays like that. Whenever it needed, it's tried to come at the place of your, at the end of your presynaptic area. It just muses and fuses itself, and it being collected over there and giving you a small hole-like structure, releasing GABA outside. And this is all the work done in the presynaptic clefts. And this presynaptic is nothing but the cleft, the presynaptic junction is nothing but the axonal part. That means different types of neurons, different types of nerves, and different types of neuroaxons complete work. So the synapses between the presynapse and the postsynapse, that is called as junctional place. What we can call it as presynaptic junctional place. And now what we can see is this is all the junaction junction, what we can call synaptic junction. And this GABA is being released into the synaptic junction so that it is being absorbed by the postsynapse, and it is being transformed to the blood, smooth muscles and to the organs, so that the inhibition of the organs will start taking place. And now, as I said before, it is being released into the presynaptical junction, and there some of the GABA will be collected by the postsynapse, and some of the GABA can be taken outside by a process called as metabolism. And this metabolism can be done. Why? Because we need metabolism so that some of the GABA which has been released outside into the presynaptical junction can be collected and stored back into the presynapse for the pre-uptake, for the pre-junction, for the storage place. How it is doing? It is done by a process called as metabolism. And now we'll see what happens to the metabolism of GABA. As I said before, the GABA has been taken by the presynaptic junction, and it is being transformed and reuptaken once again to the presynaptic neuron, and that is called as metabolism. And this metabolism is done by the GABA, which has been taken up and conformed itself into or transformed into synthesized itself into succinic acid. This succinic acid is one of the component of Krebs cycle, and this succinic acid is transformed itself into SSA. This SSA is nothing but semi-succinic acid, and this is also done by the GABA T. What is, as I said before, GABA T is called as GABA alpha oxyglutarate transaminase. It is being succinic acid is converting itself into SSA, nothing but semi-succinic acid. I wrote here onto the top. SSA is being converting itself into alpha glutaric acid, and this alpha glutaric acid is being converted by an enzyme called as SSADH. What is SSADH? Semi-succinic acid dehydrogenase. So this is one of the enzyme which is converting SSA into alpha ketonic acid. That means the metabolism of GABA is done back to the alpha ketonic acid by 3 steps. One is it is converting into succinic acid. The succinic acid is converting into semi-succinic acid, and semi-succinic acid is converting into alpha ketonic acid, and this alpha ketonic acid is converting oneself and turn back to the Krebs cycle. And this Krebs cycle is once again the compound called as alpha ketonic is collected, and once again it is grasped for the presynapse for the further process of GABA. So we can see that the cycle being started from the alpha ketonic acid, and once again it is ending with the alpha ketonic acid by different 5 to 6 steps, and the whole process from alpha ketonic acid to the ketonic acid, this is called as glucose or GABA shunt pathway. And as I said before, glucose and pyruvic is also some 2 precursors which are used for the synthesis of GABA. Why? Because the glycolysis cycle which has been produced over there are also helpful for the preparation of glycolysis cycle, and also they act as a precursors over there. So we can see that alpha ketonic acid to the glutamic acid, and the glutamic acid to the GABA, and the GABA to succinic acid to the SSA, nothing but semi-succinic acid, and semi-succinic acid to the alpha ketonic acid, and so the alpha ketonic acid once again grasps to the Krebs cycle. The whole pathway is called as GABA shunt pathway. That means we can see that the synthesis, the release, and also the metabolism of GABA over this pathway. This is called GABA shunt pathway. And now we'll see what is happening, what the released GABA over here. The released GABA over here has been taken up by the 2 receptors over there, what are called as GABA A receptor and GABA B receptors. What are these 2? GABA A and GABA B are the GABAergic neurons which have been collected for the GABA and transformation of the inhibition characters, so that it is being taken up by the smooth muscles, because the smooth muscles are winded up with these nerves, and it is being supplied to the organs so that the inhibition starts over there. And so what happens here, I want to give you a small before what happening before to the post synapse, what is happening in the presynapse. And on to the top of your surface you have 2 different types of receptors over here. This is called as potassium channel, and this is called as sodium channel, and you have another one which is called as calcium channel. So here, as I said before, the GABA is of 2 types. One is GABA A and the other one is GABA B. What is GABA A? GABA A is one of the GABA receptors which consists of 5 components or 5 subunits, what are called as 2 alpha, 2 beta and 1 gamma, which consists of 2 sites on it, what are called as 2 GABA subunits. One is called as benzodiazepine units, and the other one is called as piroxicam units, and above the piroxicam you have flumanidil units, and above this they are called as barbiturate units. So what are these 2, all of these points? These are all the points where the GABA has been collected and which is being inhibited with some conformational characters. So if you see the structure of GABA, it is just like a pentameric rose petal like structure which consists of 5 subunits. Here we can see 2 alpha, 2 beta and 1 gamma, and on top of it you have 2 units what are called as GABA units, which collect GABA and make to sit onto that receptor. Once this GABA comes and sits onto this receptor, the opening in the center is having some tube-like structure or like canal-like structure, which has been giving you pathway so that more and more chlorine are derived out. Here you can see the chlorine driving out. And one more we can say that this GABA is called, it has to cross 4 membranes of the cell structure. So here we can see once the chlorine has been released into the post synapse, we can see it has to cross 4 membrane layers of it. So it is called as, I have donated 4 times. Why? Because it has to cross 4 layers of your cell membrane. And what is the difference between GABA A and GABA B? Here GABA A is called as metabotropic actional GABA receptors, and that means it is being worked by the voltage ions. And the GABA B is called as, GABA A is called as ionotropic. Why? Because it is working with voltage ions. And GABA B is called as metabotropic. Why? Because it is acting with GPCR action. We know that GPCR is nothing but G protein coupled accept receptors, GPCR. It is GPCR. So this is the only difference between GABA B and GABA A. As I said before, GABA A is voltage ion functional receptors, and GABA B is metabotropic receptors, which is acting with GPCR, nothing but G protein coupled receptors. So the GABA B, actually we can see that more and more GABA A receptors are working in the brain structures by GABAergic neurons. And now, as I said before, the post synapse consists of 3 ions, 3 channels. One is called sodium, potassium and calcium. And onto the post synapse you have GABA A and GABA B, which is driving, this is all driving out plus ions, and this is driving out minus ions. So what happens over here in presynapse? In the presynapse you can see 2 types of changes. What is it? Whenever there is been inhibition or whenever there is emotional balance, as I said before, what happens is the sodium, more and more sodium will drive into the presynapse. And what happens whenever sodium is being driven inside, it is being making the positive ions of your presynapse. We can see here all the positive ions which are present over here. And in order to drive it out, once again, more and more calcium is also being driven out. We know that it is made up of Ca2+. So more and more positive ions are being accumulated over here. So why? Whenever there is accumulation of positive ions into the presynaptic area, that place is called as depolarization phase. Nothing but the action potential starts over here. Nothing but the electrification of your neurons making into the firing of neurons. And whenever this happens, the firing of neurons, it starts stimulating the presynapse. Whenever there is a presynaptical junction or presynaptical action potential, more and more of potassium will drive out, as I said, indicated over here. So what happens is, whenever there is a potassium driving out, there is a balance between the sodium and potassium, so that the actions of depolarization, hypopolarization will go on continuation. Once it starts firing, these synaptic vesicles will come close to the presynaptical junction area, and it is being released out, and the GABA is being released out. So whenever this being happen, the action of repolarization has to be started, so that the firing of the neuron will be stopped over there. And once the firing of the neurons has to be stopped, more and more potassium has to be driven out, so that the potassium layers which has been having positive areas will go out, so that the balance between the depolarization and the repolarization will be happen, and action of firing will be stopped over there. Now the action of postsynaptic has to be erased. So this is done by your GABA A receptors. Why? Because the GABA has been released into the junctional area, and this junction of the area will be absorbed by the GABA A receptors. Actually, I have to take it up both of them, but as so GABA A is more, I am dealing with GABA A over here. So I gave you the structure over here and already indicated what happens over here. So whenever the GABA strikes onto the GABA receptors, the area of the two top areas which is present onto the top of pentameric receptors, that area will be collected by the GABA. So whenever the GABA comes and sits there, the channel of the chlorine, here it is being indicated over here with a green color. So when GABA comes over sit here, there is many conformational changes happens over to the GABA side, and all the calcium channel will be widened so that more and more calcium will be more and more chlorine will be released out, making all the minus ions onto the post-synaptic area. And here this is called as dendritic shaft. Why? Because it is post-synapse. And so the GABA has been collecting the Cl minus ions where the chlorine ions are being indicated and minus ions are being accumulated over here. So once this accumulation of chlorine has been heightened, the inhibition, that means the firing of urines has been stopped off. Why? Because there is more and more chlorine, more and more minus ions, when the plus ions are being closed off, so that the inhibition of, that means the firing of the neurons will be stopped. And this collection of this minus ions will be transformed all to the neuronal areas which has been winded up onto the smooth muscles, and these smooth muscles are being collected to the organic areas, and that particular organ will be inhibited in that excitation. And so this is GABA is one of the more neurotransmitter which is done the work of inhibition area. And as I said, it has been acting in the limbic system, eyes, temporal lobe and amygdala, and also the positive senses, the minus ions, the plus ions, the syntactics, the release, the structure and the storage has been indicated over here. So this is a small story about the GABA, which is called as a neurotransmitter released in the brain. Thank you for this section, and we'll see in the next section. Bye.